So I'd like to invite Dr. Leo Almeida up. He is the session chair for session 1. Great, yeah. Okay, yep. Dr. Almeida is a neurologist and the movement disorder division director at the University of Minnesota, and will lead us through session 1.
All right, thank you. Yes, could we have everybody who is speaking just to come over and get mic'd, please?
Did I just steal your seat? No, you stole Michael's seat.
And while we're getting them ready, maybe I should just share the same feelings and the same concerns that Kai had. So thank you for, hey Enrico, for inviting me. And it's always a very bold move to get a clinician to be sitting amongst a lot of very well-skilled researchers. So when we got invited to put this session together, the goal was to try to be a little provocative, right, if we could, and look at a concept that everybody has been talking about for the past 10, 15 years, which is on the clinical side reinventing what we call precision medicine in clinical care, which is focused on patient-specific symptoms. Yeah, so, and that's what we do, right? So I've always been known as the troubleshooting guy, right? Since when I was on faculty in Florida. And, we collected a few friends over the years that other folks had a good amount of trouble figuring out and optimizing, and, we ended up where I am right now. So I have a lot of different bios from everybody, all these speakers here, but you know me, I never follow the rules and the protocols, so I'm just going to focus more on the reasons why they're here. So we have a number of amazing speakers here, and we know them really well personally, and once the time comes, I'll do an introduction, maybe put them a little bit on the hot seat. So again, I had bios on everybody, so who is going first? Is that Enrico?
Yeah.
All right, so we could spend the entire day talking about Dr. Enrico Opri. I'm not going to read about how successful he is, because everybody here is really good and very successful, very well accomplished. But I've known Enrico now for 10 years, right? So Enrico used to be a student in our lab at University of Florida when I was in training. And then now you're University of Michigan, right? So he's a very brilliant guy, and he's done a good amount of research with brain signals, right? If you see a lot of the work coming on closed loop that came on closed loop for essential tremor, part of the NIH-sponsored trial the University of Florida did. He's the guy who did it, right? And now he's pursuing new adventures at University of Michigan, looking at brain signals and so on. So we'll let him come over and share a little bit more about what he's doing. Yeah, Enrico, it's all yours.
Thank you. All right. Thanks so much. And it's been a long time. And as I say, they grow so fast. So now I have a small kid, and I can definitely see that actually happening. And okay. Well, first things first, thank you so much for sharing your experience and sharing your thoughts as well. I really appreciate it, because after all, that's the main driving for what we're doing this to begin with. Actually, it's great to be back here actually at the Neuromodulation Symposium. This actually is, usually I used to be on the student side, so now it's kind of nice to be on the other side as well. And so I really hope everybody is well here, and the caffeine, I hope is still strong. Actually, it was mentioned before, so I hope that's the case. So briefly, actually, so I want to talk about a particular work that I'm currently spearheading in my lab. Actually, one second, let me move the pointer. So the work is currently spearheaded within my lab. We are currently focusing on DBS, Parkinson's disease, and dystonia. So one of the most important factors is actually the correct placement of the lead within the brain in the target region of interest, and which can be subtarmal nuclear, for example, or globus pallidus internus, TXTN and GPI, basically mentioned afterwards, and together with the post-operative programming as well, because we want to provide the best symptomatic benefit with the fewest side effect. So the recent introduction of segmented DBS leads has enabled a more specific and targeted stimulation, but the problem now that we have even more complex programming sessions. So this is because it's very time-consuming and a trial-and-error process for both the patient and the clinician. So what we're proposing in this work is to have basically an objective measure that can help inform the clinician on the best setting for stimulation. They can help to validate the position of the lead before closing during surgery and decrease the work complexity of the approach. So these can be potentially achieved by usage of another biomarker, the DBS local potentials, DLAPs, or also known as ERNA, which aim to inform us on the optimal console location from the implanted lead itself. This is relevant for people undergoing DBS because, again, we want to make basically the DBS targeting, validation, and programming a little bit easier for them. So previous work has been focused on leveraging single unit or pathological bit activity, which is however affected by a variety of recording conditions and require the patient to not be under anesthesia, for example, or they need to comply with your task to begin with. And so today, both me and Dr. Johnson, both DLAP and ERNA, they are quite promising on the domain. For transparency, both DLAP and ERNA, they actually are the same thing, but more at the later. I think this is an interesting point of discussion about the naming. And so, as mentioned, the DBS local potentials, or DLAPs, have been suggested as an additional method for mapping. So they actually are evoked by delivering stimulation through the DBS lead themselves, and defined as local because typically we elicit them and record them in the nearby contacts of the DBS lead itself. So they actually be suggested they arise due to reciprocal connectivity between the SDN and the GPE. They manifest as a resonant look-like activity. As a DLAP evoked by stimulation, they can be recovered on adjacent contacts. The recording is often non-trivial in current off-the-shelf amplifiers, as many of you that work in Neuromodulation know already. But the benefit is actually that they have a larger amplitude compared to other biomarkers, such as beta, and so they're actually easy to detect. So in our work, we are collecting simultaneously from all the concepts of the DBS lead for the maximum spatial acuity, which is especially useful for segmented leads. For this purpose, we develop a couple of methods we show later, and if you can see basically here in red, this actually is the obtained waveform actually of an example of DLAP. So again, here to give an example of why these can be useful for localization, these actually are both patients that the surgeon had planned to be both SDN. Again, actually, as an engineer, I just like to see these cases sometimes because it actually help to really map out where we are. And so you can see here, basically, where we are on target, we do see basically the best potentials are appearing, regardless of where we're stimulating with DBS electrode. But actually, in this case, when you're off target, even just 1.5 millimeter away from it, we actually do basically not have them anymore. So across here, actually, we have two studies that we talk about, where we use the alpha-omega and omega system. Basically, it's a clinical system. More at the point why later. So we actually use monopolar recording, again, because specifically we wanted to not use bipolar stimulation or bipolar actually recording to make sure that we actually had all the special information we actually could get. So we actually had two protocols. Basically, one was like a low-frequency stimulation for this purpose, basically considered to be single-pulse stimulation, and high-frequency bursts of stimulation as well. And we are stimulating across all the contacts in a monopolar fashion. So the point here, basically, is that we cannot also record, typically, in clinical systems, activity from the contact being stimulated, which is a problem, because, again, we want to begin with to record a biomarker that is evoked by stimulation. So at that point, actually, at the same time, actually, we also define throughout this work a hotspot, basically, the idea that regardless of whether stimulate across the DBS lead, so you're supposed to basically get a response that localizes within the same region. And so we use that information afterwards across different paradigms. You can see here, basically, qualitatively, two different responses from low-frequency stimulation and high-frequency stimulation as well. And so, importantly, a couple of methods we developed actually is a model-based denoising for the artifact that we actually try to estimate what is really simplified model for the tissue response from stimulation, and to really try to dissect what is the artifact and what is the underlying physiology without actually having weird alterations, especially because it's useful to quantify the amplitude to begin with. And at the same time, actually, importantly, the core of this work, actually, we recovered the DLAP amplitude we estimated actually on the contact that was under stimulation, but actually which could not be recorded due to device constraints. And we fitted independent Gaussian models, basically are shown in this figure here. And so in this way, we are able to obtain back what were supposed to be amplitude at the contact that it was not actually being stimulated and not recorded. So we use this method throughout this work. And the idea here, showing here in one patient, for example, we recorded with a parallel system in a subset of patients, like around 5 with 9 signs recorded, both with an Omega for stimulation and a TT system for just recording. And the idea here is that we could actually show that our model was actually doing a good job, basically estimating what was supposed to be the amplitude at the contact stimulated. You can see here across all the sides we recorded, we actually have an R-square for both stimulation protocols around 0.8 to 0.9. And so with that said, that's actually what we use moving forward. At the same time, we're also developing hardware solutions to ensure we're actually able to record from the contact being stimulated to begin with, and solve a couple of different issues that we'll talk about more later. And actually, my student Jana, we actually have a poster about that. And so please actually go visit if you want to hear more about it as well. And so the idea here is we can actually avoid saturation problems, ringing artifacts from the amplifier itself. And at the same time, actually, we can avoid loading effects, which can be a problem, for example, in some devices, where basically we want to make sure that all the current we're stimulating actually goes to the patient, not the amplifier, because otherwise you have this beautiful plateauing effect, but actually it's not physiological, just because you actually are conducting current through your amplifier stage. And so with that said, actually, aside from the hardware developing, so going back to the data, based on the model version before, we can see qualitatively a special distribution of our DBS potentials. You can see the focus within the SDN specifically in both stimulation protocols. If we compare with beta, for example, in beta as well, actually, we still have the concentration within the SDN, but in this case, actually, it's more spread. We can see more actually later, for example, when we compare different type of measures. We actually were caring about like how does it compare to MER mapping, because again, for awake surgery, that's still the standard of care. And so we actually, again, we are clinicianally defining independently the dorsal and ventral border of the SDN based on the physiological activity we're recording. And what is important here, basically, you can see here is the ventral, and here actually the dorsal. That's what one is here, and actually have a good discrimination across the different locations within the SDN, which actually is a little bit lower in the case of the beta as well. And so these are actually promising for using, for example, depth mapping in comparison with functional mapping. So the other way actually we compared it is based on imaging. So in this case, we did it in 2 different ways. One actually with, we actually sampled projecting on average trajectory across the patients. And so again, similar to what we've seen for the MER, what we want to see basically is a good discrimination between what is the ventral and dorsal border of our structure at SDN. And important here, basically, if we just look at the maximum of these BS potential activity, we actually was maximizing within the SDN specifically. And while we were going for beta, for example, we actually have a little bit of trailing in deciding what is like the dorsal portion as well. And because if it was manifested, actually, if we look at the slice, for example, of actually across our trajectory, basically there is like a nice focus of the BSF potential activity, which is not necessarily there for beta. And we also focus on post-programming because that's why we mentioned before, evaluating how the amplitude relates to clinical efficacy. And so we basically obtained the contact effectiveness for each contact that could be evaluated. Again, actually from a blinded neurologist after 1 year optimization from the charts. So this indicates that there is actually good trend actually between the amplitude and the clinical outcomes, while actually it's not necessarily as evident in the case of beta power. There's still a trend, but it's not significant. And but the key question I want to bring here is that can we actually, so this is all nice and everything, but can we actually change the way we do surgery as well, actually try to bring functional mapping in the sleep surgery as well, without changing the pharmacological aspects of anesthesia? So the hint is yes, that's why actually I'm talking about it. And at the same time, we can record, for example, again, SDN labs, also under anesthesia, they have a different morphology, but what I'm showing afterwards is that we maintain spatial specificity. For that purpose, basically, we're going to do a different study, focusing instead on awake versus a sleep surgery. And again, this sequence can do that because we actually do implantation during the same day. Stage 1, we implant the lead, and stage 2, we implant the device. So we leverage that window to record in the same patient with the same protocol. So the nice part here actually is this work is spearheaded by my student, Emily Benz, which also have a poster tomorrow as well. And we're currently enrolling patients from both UFM and VA. And the important part is we still use both protocols just to see which one are more relevant for this mapping. And we're also doing collaboration with Washu, we call actually Peter Brunner and John Wheely as well. And so again, looking again at the distribution with awake data for reference in these different populations, we could still see that we still have the discriminatory aspect actually between the different portion of the SDN. But actually, importantly, beta, but not as significant. But the important aspect, which I found more interesting, if we go again, looking actually in 3D, we can actually see that the labs still again show this focus here, but it seems to suggest that the focus of the beta is not necessarily overlapping with what is actually found by the BSO potential, suggesting that maybe if you're looking at is actually different related phenomena actually that we can actually dissect more and further actually in the future. So, for example, here actually we can compare actually the raw values actually across the contacts in different aspects actually for actually between beta and for example and actually the lab as well. So you can see here we actually have although like a really weak correlation between the what is the low frequency amplitude and the beta amplitude, which again actually makes sense because again actually with slightly different focus and centroid actually for the we're showing in the 3D image. Which may also manifest actually in the high frequency. While instead actually there's a good correlation actually if we look just at the matching between the spatial information we get from both stimulation protocols, the low frequency and high frequency. If you actually look at the asleep portion, we actually do have that, we do still have discrimination across different locations. Importantly, actually, the beta is basically gone, which we can see even better actually if you look at the again, actually in a normative space in MNI, the distribution of the data basically is like we are not able to discriminate actually anything there, which actually is something that's being shown from past groups as well. But for comparison, instead, actually, we can actually instead have actually still a focus of TLAP high frequency and also low frequency as well, which is a bit less. So actually, you can see here actually the comparison between awake we've seen before and asleep. So interestingly, we have a less correlation, a little bit more divergence between the lab listed by different stimulation protocols under anesthesia, which brings the question about what really we actually typically use sevoflurane and propofol, and like how those actually interact actually with the network of interest. And at the same time, actually, if you look at the beta, for example, there is no actually information kept basically maintained between the two different recordings, between awake and asleep. One actually, interestingly, especially if you look actually at awake versus asleep, in both protocols, we do maintain basically a correlation, especially in the high frequency. And so that's why moving forward, actually, what we focus on is actually mainly high frequency stimulation for the purpose of recording during asleep. And so we can also record actually in the GPI as well, but Dr. Johnson will talk more about as well actually afterwards. And so the important focus actually here is that we actually basically get these focus of the ERNA DLAPs between the GPI and GPE border, but beta actually is more challenging for this purpose because it doesn't necessarily peak near the border of the GPE GPI. So when we look again at the predicting the therapeutic contacts, we notice that there is a good performance when choosing one of the top two contacts. And but most interesting, actually, what I found is that it has a really good accuracy when we compare with the effect threshold instead. And so actually, this means that this paradigm can be more useful actually to, for example, to get effect threshold in this way, actually have something else, for example, multiple potentials to try to get side effects, and combine actually could get actually much better prediction of what is the endpoint for therapeutic contacts. So going back to the rationale about why we want to use this approach to begin with, so how it could be applied to clinical implantation. So here you have the same patient recorded both awake and asleep. So we should show an interesting match. So we could be developing an algorithm that basically prompts the clinician but actually with the depth information, so in this way, actually, you can just like know where they are in space. And you can see if it correlates with what's the imaging actually post-operative as well. For that purpose, we actually are focusing on a few efforts to get actually also with which actually does in group, and that's why collaboration with Washu, with Brunner as well. And so we'll actually integration with Research Dozen for the Neuro Mega system. And so this really is to basically enable real-time recordings and computation of the DLAP markers. In this way, actually, we can actually have more useful paradigms for this purpose. So you can see here an example of a paradigm spearheaded by my student Prarana, which also will have a poster tomorrow. Specifically in this case, where we obtain actually after running the algorithm automatically, it gives actually depth information. So in this way, we know if we're supposed to push or not the electrode in different locations. So at the same time, actually, we interest actually in tools. So interest actually, how can we make this even faster? So for that purpose, we're developing basically new denoising routines as well, aside from the one shown before, basically actually based on convolutional neural network. And so the purpose here actually is to actually have a much major speed up on the computation. So right now, actually, for the previous one, actually, it usually takes around 1 second for 30 DLAPs. In this case, it takes around 20 milliseconds for 100 of them. So we're actually much faster actually in getting those. And the idea again, actually, we got to remove the artifact without keeping the neurophysiology. And so for that purpose, also we tested with some controlled data with general tremor patients, which we know do not have necessarily DLAPs or ERNA. And that's why it's good to see that we don't have hallucinations, basically generating DLAPs when they're not there. So we're going to purposely some examples of like what does it mean for using in the clinic. Actually, if you have a lead implanted shallow before closing, the idea actually that you will actually have this response here, and you're able to actually push electrode in location and basically correct it before closing the patient, which again is more awkward to do actually afterwards. But interesting question, basically, instead of just focusing on that information, can we leverage truly spatial information in 3D? So basically what we did that, for doing that is we divide actually our sample space in boxes by 2 by 2 by 2 millimeters, having at least 3 different DLAPs within the same voxels, and with information to train our encoder classifier architecture, which then basically we use the one we saw before for the noise in the data, but with these actually instead we predict basically try to estimate both the location, basically the 3D coordinates, at the same time actually they label the voxel that belong to. And so this really leads to really interesting performances. Basically, show really well above chance level accuracy, which is around 4.5%. And we can also appreciate for like body vision metrics. And so developing the model basically to show what is the predicted lead location, which is shown in red here, compared to the known location actually here in green. So that's actually something that we're working on, and that's what specifically my student Prarana will actually show tomorrow. And actually, and so with that, actually, the purpose actually is we can actually give directly what the lead was supposed to be. And one thing I want to leave with is, at the same time, the product question is the lab to begin with a pathological biomarker? So collaborating actually with Anel de Vernier and Marco Nolli, actually at Emory University, we actually did some recording in human primates, and actually we saw actually the lab there as well, which actually I believe that your lab saw that as well. And so the point is that we bring the question is, this actually is not necessarily present due to pathophysiology, but actually could be present there actually just because of the brain structure itself. So at the same time, actually could be affected by it. So I think this is a good question to start to pursue. And so with that, actually, I hope to show you how we can actually leverage this marker and for both localization and programming as well, with some techniques of how can we do better recordings and faster estimation. And this work actually was possible thanks to patients that actually dedicate their time. Otherwise, this literally would not be possible. And I also want to take a collaboration with the Neurology and Neurosurgery department, my colleagues at U of M, Washu at Emory, and all my lab, actually, the part of it is here, and which we like to have sometimes some fun photos and as well, and the foundations as well, and NIH as well. Thanks so much. Question afterwards? Questions afterwards?
I think we're going to do questions after, right? Yeah.
Okay.
So I realize I'm too short and I can't, if there is a timer here somewhere, I just can't see it. I keep stretching myself. So, but I think, so there are a couple of interesting questions, but we'll save that for after everything. So I think next we have Kara, right? I've also known Kara for a long time. So Kara has been, in addition to the Florida group, coming together with Chris Butson, right? When he came from University of Utah. So we drove Kara crazy quite a few times, you know, because she was put in charge of analyzing a lot of data from the International Tourette's database, and the main hub is at University of Florida, but the imaging was being handled by Chris Butson. But then, you know, maybe we burned her out a little bit. She decided to kind of shift gears, right? But I've never worked with somebody as organized, you know, as a student, because everybody, you know, students are always, you know, a little all over the place. No offense, but
It's true though, yeah.
Right, yeah, right. But Kara's been amazing, and a lot of the pipeline that Florida uses right now is thanks to Kara. To helping organizing everything, and then she's going to present some data on, I guess, I don't know if it's DLAP or ERNA, right? You guys are going to have to decide that.
Names, yeah.
Many
Going to
names.
have some sort of like,
Yeah.
tortilla fight after this to
No,
see
I mean, like, it's-
what's going to happen. But, you know, Kara's going to present like the GPI side of the story with some more data to show to us. So, Kara?
Thank you so much for that introduction, Leo, and thanks for the invitation as well. I'm really excited to be here. This is my first time at this conference, so it's great to be here, and I'm excited for the rest of the sessions as well. So I'm going to be continuing on in the same topic of evoked potentials in deep brain stimulation, but I'll be specifically talking about these evoked potentials in the globus pallidus, or GPI, in both Parkinson's disease and some of our latest work focused on dystonia as well. So I won't go into too much of the background. Enrico did a great job of introducing this problem of trying to optimize deep brain stimulation therapy and identifying effective stimulation parameters. Because we know that the therapeutic outcomes of DBS rely very heavily on being able to deliver stimulation to very precise deep brain regions. And how we can kind of titrate those regions that are being stimulated is through selection of these stimulation parameters. So the contact that's used for stimulation, as well as the amplitude, pulse width, and frequency. And these numbers of possible combinations that we can choose from are only exponentially growing with these directional leads, and now with adaptive stimulation paradigms, different stimulation patterns as well. And so this really makes this a complex process in order to figure out how to best deliver this therapy. And we know by titrating these stimulation parameters, both intuitively and from computational modeling, that changing these stimulation parameters changes the field of stimulation that is delivered in the brain. But unfortunately, despite this large parameter space that we have to transverse, the current workflow for DBS is largely trial and error still. So the patient undergoes their implantation surgery, and then they come into the clinic for months of iterative processes where the clinical care team tries to converge on the most optimal stimulation parameters. So this is really a great burden on the patients, their caregivers, as well as the clinical care team. And this is largely a trial and error process still, because there's still a lot that we do not know about DBS and how to best optimize this therapy. So this includes specifically where do we need to stimulate on a patient-specific basis. We know generally what target region we need to implant in, but we don't necessarily know how to hone in on the precise spot to stimulate, as well as the downstream networks that need to be modulated. How to stimulate, so I mentioned all of these different parameters that we can titrate, but we have patterns of stimulation now. We can do even more complex things, but we don't really know how to use them yet. And then, of course, what activity are we trying to modulate within the brain? So we generally know these biomarkers, beta power, for example, that is associated with symptoms, but on a patient-specific basis, how do we use this to drive the therapy? And to make matters more complex, all of these things are definitely interrelated, and we don't really understand those interrelationships very well either. So this really drives the need for data-driven and more personalized approaches to guide DBS and make this a more efficient and effective therapy for these patients. So at the University of Florida, we really try to take advantage of this very unique opportunity that we have to record neural activity from deep inside of the brain in patients who are undergoing DBS therapy or DBS lead implantation surgery. So very fortunate to be able to collaborate with a large team of neurosurgeons, Kelly Foote and Justin Hilliard, as well as our entire neurology team as well in the OR. Using the alpha omega system, we're able to record local field potentials from the DBS electrode when it's being implanted, and we're able to stimulate and record neural signals from each of those electrodes on the DBS lead. And so we've kind of developed this workflow using semi-automated recordings where in every single patient who undergoes DBS lead implantation, we try to record at least a resting state recording just a minute before any stimulation has been delivered. We also record during symptom assessment, both on and off DBS, and we also collect EMG to try and measure those movements as well. And then customized protocols, such as evoked potentials, which I'll talk about. And we try to limit these to 2 to 4 minutes, where we can very quickly acquire them, basically press a button and have it run this protocol and collect these recordings. So this has really allowed us to build up a large data set at this point of many patients who are undergoing DBS. And so we have several ongoing projects using this data set, but in particular today, I'll be talking about evoked potentials. So specifically, I've become very interested in this potential biomarker, called evoked resonant neural activity, but also called all of these other things as well. I think eventually we will converge on a single name, and we talked about that. I think we all need to have like a vote or something on what it should be called from here on out. But it's the same thing as the DBS local evoked potentials that Enrico talked about. There's also a new version of ERNA using recurrent instead of resonant, which I think I would vote for that one at this point, but to be decided. So again, this is a high-frequency oscillatory pattern of neural activity that is elicited by the DBS, and it gets this name resonant or recurrent because of this characteristic pattern where it starts with a high amplitude peak and successively decays back down to baseline after the DBS is turned off. And the current hypothesis is that this signal arises from modulating reciprocal connections between the subthalamic nucleus, or STN, and the globus pallidus, the GPE and GPI. And so stimulation in either of these targets elicits this ERNA response. And there have been several studies now that have shown that ERNA seems to be associated with the site of therapeutic stimulation, as well as potentially the stimulation parameters that should be used for chronic therapy, which Enrico nicely showed in his talk. But all of these studies that I've listed down below have all focused on the STN, which is the most common target used for DBS and Parkinson's. But as some of you may know, University of Florida is a very GPI-heavy center, and we take pride in that. And so we took advantage of that and tried to characterize ERNA within the GPI. And so in this first study, with the help of Janine on a lot of the clinical aspects of this work, we delivered bursts of high-frequency stimulation with intraoperative recordings of Parkinson's patients who are awake and off medication undergoing DBS implantation surgery. So we deliver these bursts of high-frequency stimulation with the idea that we can sort of elicit this response, but have time in between the bursts in order to measure it. And then we repeat these bursts several times from each of the contacts on the DBS lead sequentially. And so the purpose of this study was really to establish whether ERNA may be a useful biomarker to guide GPI DBS. And so the first question that we asked was, does it vary depending on what contact is being stimulated? Because this would be important. We need to have differentiation between or across contacts in order to use it to guide programming, for example. So here's one example patient where you can clearly see stimulating from contact 2 here in red elicits a much higher amplitude, more robust ERNA signal compared to the contact in orange. And this is pretty variable across patients, in terms of the ERNA response, as well as depending on which contact is being stimulated. The second question that we asked was whether ERNA is spatially localized, which would be important if we wanted to use this for something like intraoperative mapping. It needs to follow some spatial trend in order to be able to localize where you are in the structure. And so what we found is that stimulating at the border of GPI and GPE or within the GPI was associated with a significantly higher amplitude ERNA response. Very consistent to what Enrico briefly showed. And then finally, importantly, we performed a retrospective analysis where we determined whether ERNA may be correlated with the stimulation parameters that are used for chronic therapy. And so we compared the contacts that elicited the highest amplitude ERNA in each patient to the contact that they were programmed after months of iteration with their clinical care team. And we found with an accuracy of about 70%, ERNA was able to predict what contact should be used for therapy. So overall, this study really showed that GPI-ERNA is indeed a promising biomarker to guide DBS in PD. But in this study, patients were only implanted with this conventional quadrupolar DBS lead. And as we all know, we are increasingly living in a directional world where many patients, if not all patients at this point, are being implanted with the directional electrodes. And so in this next study, we wanted to know whether ERNA may be useful to guide directional DBS. So we recruited 60 patients with PD undergoing either GPI or STN DBS, about half and half in this cohort. And we delivered the same burst stimulation protocol, but instead we delivered these bursts of high-frequency stimulation from each of the directional contacts separately and recorded the ERNA response. So we could compare, depending on what direction was being stimulated, how this ERNA varies. So here's a couple of examples here with directional stimulation. And we first wanted to validate our localization in this new cohort, given it's a new electrode, as well as we're delivering directional stimulation rather than this ring mode stimulation. And here you can see the contact locations across all of the patients mapped with the respective ERNA amplitude that was elicited. And if we interpolate those onto a heat map, we found a nice correlation again where stimulation within the GPI or at the GPI-GPE border was associated with a significantly higher amplitude ERNA response. And this corresponded nicely with our previous work. In the STN, we also found a nice localization where stimulation within the STN elicited a higher amplitude response, but particularly localized to the dorsolateral part of the STN, which is really exciting because that's hypothesized to be the therapeutic sweet spot within the STN. So this corresponds nicely with Enrico's lab's work, as well as a recent study by the group in Toronto that has shown a nice correlation with this sweet spot. On an individual level, there seems to be some directional tuning of ERNA, so it varies quite a bit across individuals and depending on what direction is being stimulated. So here in this GPI example, you can see stimulating in this sort of posteromedial direction is associated with the highest amplitude response, whereas here in this STN example, stimulation in this more anterior direction, but particularly on this second level of the electrode, is associated with a higher amplitude response. And at a group level, we saw a lot more variability within the GPI. There wasn't a super clear tuning pattern of particular directions that were associated with this higher amplitude response. But in the STN, we see quite a striking pattern where stimulation in typically this more anterior direction or posterolateral direction seems to be more commonly associated with a higher amplitude response. And so we hypothesize that it may reflect targeting strategy in the STN, where leads tend to end up more on the medial side of the STN with the goal of avoiding the internal capsule because you don't want to have too many side effects. And so if you're on the medial side of the STN, then stimulating in the more anterior direction would be pointing towards the dorsolateral STN, or at least towards the STN. So this is really exciting. We're working on sort of bringing this directional aspect with a bunch of arrows in 3D space into our heat map analysis. But so far it's looking really promising that stimulation towards these hot spots seems to be associated with this ERNA response. We've also been exploring whether ERNA may be useful to guide stimulation parameter selection beyond just contact selection. So we have some protocols now that we've been running where we vary the stimulation frequency that is delivered. And again, we kind of see these individualized tuning curves where ERNA varies depending on what stimulation frequency is being delivered. At a group level, it does seem like stimulating at higher frequencies may elicit a higher amplitude response, but there is enough variability there across patients where potentially ERNA may be useful to guide what stimulation frequency may be most useful for engaging this basal ganglia circuit at an individual level. And then we've also been varying the stimulation amplitude as well. And so again, we see kind of these patient-specific curves where the threshold at which ERNA is elicited varies substantially depending on the contact that is being stimulated. So here this is with directional stimulation. So you can see some contacts where it takes 5 milliamps to elicit a response, whereas others it takes just 2 in order to elicit this high amplitude response. And so we can also quantify this at a group level, and we see a difference in this ERNA threshold across targets. So the GPI requires a higher amplitude of stimulation to elicit this ERNA response compared to the STN. So this may reflect the target size, of course. We may just need a lower amount of stimulation in order to engage STN, which is a much smaller structure than in the GPI. And then we also compared with the stimulation parameters that were used for chronic therapy, and we found that this ERNA threshold was within 1 milliamp of the clinical amplitude that's used for chronic therapy in 70% of patients, and within about half of a milliamp for 60% of the patients in our cohort. So this suggests that ERNA may be helpful for determining which contact and the amplitude at which you should be stimulating for chronic therapy. So really exciting results thus far here in Parkinson's disease. A natural question, I guess, too, is whether ERNA may be useful to guide DBS in disorders beyond Parkinson's disease. So I think a natural question is dystonia, because DBS is also targeted to either the GPI or the STN to treat dystonia. And so just a little bit of background of what is out in the literature thus far on ERNA in dystonia. So there's one really nice study that has shown ERNA with STN DBS in dystonia in a pretty good-sized cohort that has shown a nice localization, again, to the dorsolateral part of the STN. There's a nice case report also showing that ERNA can be elicited with GPI stimulation, and this ERNA in this dystonia case seemed to predict which contact should be used for chronic therapy. And then we also recently published a case report in a really interesting case where we had an opportunity to stimulate and record from both GPI and STN simultaneously. And so we were able to kind of reconstruct this circuit where we could stimulate from one of the target regions and record ERNA from the other. And so this was really exciting because we kind of replicated that it does seem to involve engagement of this STN GPI GPE circuit. So we built on this work in a small cohort of patients with dystonia who were undergoing GPI DBS implantation surgery. So the vast majority of them had cervical dystonia, but we did have one generalized patient, and then a couple of those were classified as having Mage syndrome. We delivered the same stimulation protocol, so these bursts of high-frequency stimulation, and we found that indeed ERNA is robustly elicited with GPI DBS and dystonia. All of the patients exhibited this characteristic response. And we found that the responses were about a similar amplitude to what we saw in Parkinson's disease. We have ongoing work to compare ERNA in Parkinson's versus dystonia that seemed to suggest that potentially the frequency of the ERNA response may be lower in dystonia compared to PD, but exhibit a similar number of peaks. And again, we saw quite a bit of variability across individuals in the amplitude, frequency, and morphology of the responses. Similarly, we also found that ERNA varied significantly depending on which contact was being stimulated at an individual level. So we can see variability in not only amplitude, but we also saw quite a bit of variability in the frequency of the response as well. I would say even more variability than what we have seen in Parkinson's disease in the past, and this may be interesting given the next result that I'll show on the next slide. We performed a preliminary analysis of the spatial localization as well, but this is a pretty small cohort, so it's kind of difficult to build up that heat map with only 9 leads. But so far, it suggests that ERNA does seem to be localized more to this GPI-GPE border, potentially pushed more into the GPE here in dystonia, but this may be just be part of the targeting and the small cohort. So more on that, hopefully to be determined. Another question that Enrico brought up is whether ERNA may contain pathophysiological information. So far in Parkinson's, there really haven't been a whole lot of robust correlations between ERNA features and symptom severity, for example. But we decided to explore this in dystonia, and interestingly, we found that the frequency of the ERNA response was significantly correlated with the cervical dystonia symptom severity measured by the Twister's scale. So in this correlation, the lower the ERNA frequency, the more severe the patient's symptoms were. And so when I heard low frequency, that made me think about other biomarkers that have been explored in dystonia, particularly low frequency power, theta alpha range typically. And so we performed an analysis where we used our resting state neural recordings from these patients that we also acquired, and we computed the power in each of these frequency bands. And indeed, we found that the ERNA frequency is also significantly correlated with the alpha power in the same direction. So the higher the alpha power, the lower the ERNA frequency. So this suggests maybe there's something about this circuit that is sort of resonating at this lower frequency that precludes it to respond to stimulation with a lower frequency in dystonia patients, particularly in these cervical dystonia patients. And then, as correlations, I guess relationships between these correlations, the alpha power was also significantly correlated with their Twister's cervical dystonia scale. So overall, we can't really say, causal relationships between these variables, but there does seem to be some interesting interplay between ERNA frequency and this alpha power and symptoms. Lastly, we performed a retrospective analysis, again, comparing the ERNA to the chronic stimulation parameters after 12 months of optimization with the clinical care team. And we found that in all but one, ERNA seemed to be associated with the contact that was used for chronic therapy. We did have some sort of more complicated settings here, so some double monopolar, a couple of interleaving settings as well. But overall, this suggests that there does seem to be some correlation here between ERNA and chronic therapeutic stimulation parameters, which I think would be particularly impactful for a disorder like dystonia, where often there is a lack of acute response of stimulation within the clinic. Sometimes it can take months for the stimulation to really show its therapeutic effects. And so if we could have an idea of at least a place to start of which contact to program on when the patient comes out of the OR, then I think that could be very beneficial to expedite that process. So overall, our work suggests that ERNA really shows a lot of promise as a potential marker to guide DBS targeting and potentially stimulation parameter selection in both Parkinson's disease and dystonia. It's really kind of a fun biomarker to study because you get to sort of localize this circuit by probing it with stimulation. And so you can see the direct effects of changing the stimulation parameters on the evoked response. We see it's quite patient-specific with a wide dynamic range in that it seems to be tuned to specific stimulation parameters at an individual level. So I've shown here, depending on what contact and direction that's being stimulated, as well as the amplitude and frequency. We haven't varied the combinations of parameters yet, but I think that's the next step, but it gets really complicated really fast, so we're taking our time with that. We've also shown that it correlates nicely with post-operative stimulation parameters with roughly 70 to 90% accuracy in our studies and others. So there's really some potential utility here for being able to prospectively predict stimulation parameters to hopefully expedite this programming process. Still to be fully determined is whether ERNA correlates with symptom state directly. So there's some work that has shown that ERNA varies depending on the dopaminergic medication state, as well as asleep versus awake with some of Enrico's work and others. One study has shown that ERNA may be useful for decoding sleep stages as it varies in amplitude depending on the stage. But we really need some work showing real-time correlation with symptoms if we were to want to use ERNA for something like closed-loop DBS, which is something that is part of the conversation. And then finally, pretty much all studies at this point have been retrospective in nature of looking back at stimulation programming data. And we really need prospective studies now in order to figure out the best way to integrate this signal into the clinical workflow and determine its true predictive accuracy. So with that, I'd like to thank all of the members of our lab and our trainees who have been so helpful with the data collection and clinical aspects of this work. All of our collaborators on the neurosurgery and neurology side as well. Funding sources, so this work is funded by the Parkinson's Foundation, the Fixell Institute, and Tyler's Hope, as well as the NIH. And I put my contact info here if you'd like to reach out offline as well. Thank you.
Thank you, Kara. I think we're going to save questions for the end.
Great.
I will say that, on the clinical side, right, the battle has always been, physiology versus anatomy, right? And, and each one of them has its pros and cons, right? So we've had 2 talks about physiology of the target and how that correlates with, outcomes and electrode and so on. There will be some questions, I'm sure, for the end. But last but not least, we want to get Nandita up here to the podium to present some work now on the other side of the story, right? So I will say that Dr. Vitek kind of stole my thunder, talking about the new advancements in imaging and Illumina and so on, because it is part of what we talk about always, right? Physiology, it's very important, but it doesn't really tell you the distance to borders, right? And the last thing that you want is to have an electrode that is not programmable, right? Because that's a big limitation. So you can't turn it on, you can't increase settings and so forth and so on. So we need to really dial in on understanding distance to borders and figuring out ways to predict that the best we can before we close the case, because that's really a big marker between having a very successful response to the therapy versus not, right? So we wanted to get somebody who was really like an expert in the field to come in, and Nandita, Dr. Nandita Rajmani came highly recommended by Andy Horn, who is a good collaborator. We want to hear towards some of the work that she's done together with the lab there at Harvard towards personalized neuromodulation. So Nandita, take it away.
Yeah, thank you so much for inviting me. Thank you to the organizers and also for the introduction. I also realized by sitting in this panel, I don't really need to introduce my talk. Everyone has already spoken a lot about the need for optimization of deep brain stimulation outcomes. And this is a question that we've also been interested in, and I hope that I can give you the imaging take on this very important question in the field right now. So I'll start off by talking about a fact that we all very much are well aware of, that Parkinson's disease has many symptoms, and it's a very heterogeneous disorder with some patients exhibiting a lot of tremor, that is hyperkinetic symptoms, and some patients having more hypokinetic symptoms. This, of course, is also not even taking into consideration the non-motor and cognitive effects of Parkinson's disease, which drastically reduces the quality of life for patients. So how do we optimize DBS for all these symptoms? So when we look at treatment for Parkinson's disease, of course, we look towards deep brain stimulation, which at the group level is truly a gold standard for the treatment of PD. As we've all spoken about for years, there are many randomized control trials showing the efficacy in treatment of especially bradykinesia and tremor symptoms. But when we look at the more hard-to-solve symptoms, like axial symptoms, gait, and at the single patient level, DBS can lead to suboptimal outcomes despite optimal placement of electrodes. And so why could this be? One theory that we have is that DBS has long had a one-size-fits-all approach, and essentially this means that we target the same focal region to treat all symptoms at once. Of course, this has clinical validity, it has helped a lot of patients, but maybe it's time for tailoring electrode placement and programming to individual patients' symptom profile to increase the fraction of top responding patients. So that's what I would like to talk about in this talk. Of course, we are by no means the first lab to think of this brilliant idea, but there's already been work starting from the 1950s. Hassler, who was a prominent member of the German School of Stereotaxy already in the 1950s, performed around 600 lesions to propose that tremor control involves fibers that project from the cerebellum to the motor cortex. And this is in contrast to achieving optimal control of hypokinetic symptoms like rigidity and bradykinesia, which involved regulating a different network from the pallidum to the pre-SMA region of the cortex. And more recently, researchers have been trying to include personalization in both their targeting and programming. I mean, we already heard from the electrophysiology side of it. From the imaging side of it, there's a really interesting work by the Maber group in 2018, where they looked at their trial, the SEC-DBS trial for treatment of major depression. And they found that all the responding patients in that trial seemed to have 4 white matter bundles. That is the ulcerat fasciculus, the forceps minora, the cingulum bundle, and frontostriatal fibers, which intersect in their target region, that is the CG25, cingulum 25, Brodmann area 25 region. And so they thought, okay, can we use this connectomic blueprint to treat patients? Can we use this as a target instead of just the focal region? And so they enrolled 11 prospective patients, and they scanned them with diffusion imaging prior to surgery, and they found these 4 white matter bundles and targeted this connectomic blueprint across all the patients. They had 11 patients, so it's a small study, but the patients had, around 9 patients had remission at the 1-year mark. And they also tuned their stimulation parameters to find their best contact at the contact which maximally overlaps with these 4 white matter bundles. So this we call as a template matching approach. They don't call it that, but then we call it that in the sense that we have something derived from clinical observation, and this is being used for targeting. And so, of course, there are lots of studies which don't have such a connectomic blueprint. And so we look towards another example by Kernan et al. In 2020, where they looked at literature. And we know for a fact that the dendrorubrothalamic tract is involved in regulation of tremor. So can we scan this particular pathway and then use this as a target for treatment of tremor in patients? So there hadn't been that much statistical evidence. So these authors enrolled 36 patients who were of different tremor origin. So Parkinson's disease tremor, essential tremor, multiple sclerosis, and several other tremor-based diseases. And they targeted either the subthalamic or the thalamic projections of the DRT-T tract, which was also a very successful trial. Now, these are really great studies for its time, but there are some limitations of such a template approach. The first one being that diffusion imaging, especially used in clinical settings, has a lot of noise. And so a lot of these individual differences can be attributed to noise rather than true signal. And of course, from a practical point of view, this is a very costly endeavor to scan all the patients with diffusion imaging. And so probably the resulting cohort sizes would be small, and also pooling data across centers would not be so straightforward. There's a lot of data heterogeneity due to different scanning protocols across these centers. And let's take, for instance, that we have been able to map all the pathways in the human brain. That's a great point in history, in the future. But even if we are able to do that, how do we associate specific pathways to be beneficial or detrimental? What about pathways where we don't have idea about what it does? So for instance, Chris here said that he has a lot of habituation issues or memory issues. We don't know pathways for that. So how do we go from there? So for our work, we attempted to solve this in a normative way. That is, we stuck to a common template space. We took a data set of 129 patients who had undergone subthalamic DBS for Parkinson's disease, and we built a symptom-associated model for which we need an atlas. So I think Dr. Vitek already spoke about the importance of pathways and atlases. And so this is a manually curated atlas where, in collaboration with Dr. Middlebrooks, and essentially this contains all the projections to the STN or passing through the STN, something that STN is implicated in. And this it also contains the holographic atlas from the McIntyre group, some of the STN GPE tracks that are really influential in our study as well. So with this as a sort of idea of all the pathways that are implicated, probably implicated in Parkinson's disease, we used a fiber filtering approach. This is something that we developed in LeadDBS, which is an open source software that our lab does. And this concept was introduced by Lee et al. In the Nature Communications paper in 2020. Essentially, it's a very simple concept. So think of a streamline from this connectome that I just showed you, passing through a bunch of electrical fields of patients. So we have top responders and we have poor responders. These people have done well in DBS surgery. So if this streamline passes through top responders and avoids the poor responders, we can call this essentially as a beneficial tract. Now, if another tract passes through poor responders and sort of evades the top responders, this would be labeled as a detrimental tract. And finally, there's also a scenario where some tracts pass through both top responders and poor responders. Sorry that the text is in white. I somehow didn't see that. But in this case, we would just say that it's an irrelevant tract and we'd just filter it out. This is not really useful for us. It's not giving us any information. So now when we did this across all the patients and across all these streamlines that we have in our connectome, we correlated. Now the response variable for us was actually the symptoms of Parkinson's disease, the 4 main motor symptoms. So that's bradykinesia, rigidity, axial symptoms, and tremor. So we were able to construct a sort of symptom library, an atlas where you can say that these are the fibers that maximally correlate with improvement in bradykinesia, rigidity, axial, and tremor. So this was really exciting for us already because it shows a really interesting gradient. It's also not new in the sense that we have had some literature that already talks to us about, backs this up. So we had this anterior-posterior gradient at the subthalamic nucleus level with rigidity tracts projecting from the frontal regions of the cortex to the anterior region of the subthalamic nucleus. And this was in contrast to maybe the tremor tracts, which projected from the motor cortex to the posterior region of the subthalamic nucleus. And I think some of this is, as I said, has already been showed previously in literature and also in a lot of clinical observations. Clinicians usually tend to program more dorsal contacts for tremor and so on. So to summarize, we've gone from a patient level to a tract level, but to really get the value of such a symptom-specific model, we need to go back to the single patient level somehow. So what do we do for that? Consider that a patient comes into your clinic and says, I have a lot of tremor. Somehow these programming settings don't really work for me. Can we relook at the settings? So in this case, we know from our tract model that tracts which are associated with improvement in tremor are projecting from the motor cortex. So if you're able to somehow reverse engineer this tract model to find a stimulation volume that will maximally engage the tremor tracts, then that's a possible solution. And that's what we did with this algorithm called ClearTune, which is similar to the ClearSpec that Enrico spoke. I didn't realize that. But so essentially, ClearTune is a surrogate optimizer-based algorithm. And all it does is it suggests intelligently some stimulation parameters. That includes current at each contact. Right now, we don't look at frequency. These are all really interesting parameters that we can work on in the future. But for now, it looks at current at each contact. And it calculates for each stimulation volume configuration the overlap with the symptom tract that is most bothersome to the patient. And it outputs a winning VTA whenever this overlap is the highest. So we call that as the suggested setting. And so we tried this in clinics, and there was a patient who we programmed in Würzburg, and this patient had a lot of bradykinesia and axial symptoms, and you can see that the clinical symptoms already helps them a lot, right? So the image here that I have shown actually is thresholded by the tracks that the stimulation volume maximally engages with, and that already led to a 63% improvement. But can we do better? That's the question. And so with ClearTune, we could, you can see that the stimulation volumes are not that different, but they are specific for each hemisphere. And we could really push this improvement in this patient to around 88%. So this was a big first win for us. And this is just a video of, hope that works, yeah, of how this algorithm really works, right? So it's looking at different stimulation parameters throughout the iterations, and the goal is it needs to engage the blue and the yellow tracks. That's what is bradykinesia and axial tracks in our model. And finally, it chooses one that engages that maximally. If you remember, the starting was the red rigidity tracks, but this patient didn't have much rigidity, so the ClearTune here worked well. So, sorry about that, yeah. So now, but this is a patient who had 2-3 different symptoms. But what if we, for the same patient, weigh each of the symptoms specifically? So we weighed bradykinesia separately, rigidity, tremor, and axial separately. And you can see that ClearTune provides different suggestions. So you can see that in the rigidity, it really engages the rigidity tracks, and as opposed to maybe in tremor, where it engages more of the tremor tracks and less of the rigidity tracks. So one of the symptoms that I'm really interested in is also one that is notoriously hard to fix with STN DBS, and that's the axial symptoms. So something that we thought with our model, now that we have this model and ability to refine tracks to specific for symptoms, can we now find something that is specific for gait as opposed to just the axial symptoms? And so we did just that. We used our symptom-specific model to tell us what kind of tracks may be specific for gait as opposed to axial. This would be a clinically very relevant question. And so we found that the gait tracks, which is also backed up in literature, is maybe mainly projecting to the PPN. And PPN is already a target for gait. It's a bit controversial, but this is something interesting that we got. And then we also saw that there are some STN-SN tracks which are more specific for axial as opposed to gait. So now, how do we apply this in clinics, right? That's the important question always. And so at the Mass General Brigham, we had a patient actually ask us if we could use ClearTune because he had a lot of improvement with tremor, but not so much with gait. DBS really helped his tremor, but unfortunately, he had a lot of gait side effects, and they were going for years trying to program a little bit differently. And so we used ClearTune to suggest something specific for gait, and you can see that the stimulation configurations it came up with are kind of engaging these yellow tracks, which is the gait here. And they also engage these STN to PPN tracks. And my advisor calls this stimulation configuration a barbell configuration. So, and this really helped them. This helped the patient a lot. And that's the important thing, is they reduced their axial symptoms. It's been around 3 months, and I think we need to have a long-term study to really show real validity. Gait especially is very responsive to placebo. But we're really excited to see if this is something that we can do specifically for gait programming. Of course, beyond Parkinson's disease, there is a lot of neuropsychiatric diseases which are very quickly becoming important, solved by DBS. And can we use ClearTune for diseases beyond Parkinson's disease? So that was the next question that we're asking. And my colleague, Barbara Holunder, along with my advisor, is working really hard on finding this sort of OCD subsymptom model where they segregate, much like we did in Parkinson's disease, they segregate OCD into the subsymptoms and tracks that are correlated with improvements in depression, anxiety, and compulsions. So we use ClearTune, and we wanted it to just at the beginning at least, engage the global functioning tract, that is the quintessential OCD tract that passes through the ALIC. And so you can see here that it's kind of avoiding a lot of the tracts like it did in Parkinson's disease. And in the end, it kind of finds, oh, okay, this is the OCD tract, and it finds a configuration that's really involved in improvement for specifically the global function. That is the entire OCD thing. So with this, I just want to end with our vision for DBS. And DBS right now is a one-size-fits-all approach, which is valid. There have been lots of clinical improvements with this DBS model. But maybe with the ever-increasing parameter space in directionality electrodes and 16 contact electrodes, and also our understanding of how different pathways respond to stimulation, maybe we can envision more precise stimulation where there are two separate configurations, one of them engaging a tremor tract with a high frequency setting, and one of them which is engaging an axial tract with low frequency setting. So this is models like these, I think, can really help in achieving the future of DBS. With that, I really want to thank the people who gave us this data and their expertise and knowledge. We had a lot of validation data and also collaborating across continents, which was a really great experience. And but last but not the least, I really want to thank the patients who helped us, without whom this study would really not be possible at all. I also want to thank my lab, and with that, I'm excited to answer your questions. Thank you.
Thank you again, Nandita. Yeah, it's a great talk. I mean, I tell the fellows all the time, like, cookie cutter works until it doesn't, right?
Yeah.
And then you have to start thinking outside the box, right? So I think this is a great example of some of the stuff that we're getting, like new technology, new hardware in the brain, and new programming options in clinic. And, and sometimes you just have to get creative with activating different contacts in different directions and coming up with crazy stimulation fields, but that's what works, right? Because oftentimes as we spending time programming clinic, we see that, well, there's this one contact that this gets better, but it doesn't take care of everything else. So sometimes I have to do multiple activations across it. So this is great to see there's a lot of progress in that front. So we want to take some time, and I don't know how we're doing on time, but do we have time for questions? Okay, we got 5 minutes. 5 questions, 1 minute each.
Thank you very much for a wonderful session. Pretty exciting to see how we are going into personalization of healthcare in Parkinson and movement disorders. I do have a couple questions, one for Cara and another for Enrico. And the fiber tractography presentation was wonderful. I just don't want to give a space for others as well. So, Cara, I was really, truly excited to see a correlation between the frequency of ERNA and the strength of the condition in these tonic patients. So I will ask you what probably you might be doing or might be thinking, and I am curious about it. And it is, if you have a potential biomarker for a strength of the condition, you can be thinking about a closed loop system and using the frequency as the biomarker. What are your thoughts about it? And I don't know if I wait and then I ask Enrico or...
Sure, I can answer, and then maybe that'll be more simple so we don't have to remember our questions.
But I also want to know, what are you doing about it?
Yeah, so this is a really exciting result. I totally agree. And it's actually a very new result. So we're just kind of at the beginning of thinking about how we might use it. Closed loop is definitely in the discussion. I think one challenge, though, is with current devices, we don't have the sampling frequency to sample ERNA. So this is a very high frequency oscillatory signal. And so it's on the horizon, but I guess not immediately translatable yet. I think programming is an obvious place to start, definitely in more prospective studies. And I think, like I said, in dystonia, I think it's particularly impactful given that it's a very difficult disorder to find effective stimulation parameters quickly. So also with a grain of salt, this is a pretty small cohort of patients, and so we're working on the next validation cohort to see if it holds up. But so far, it looks like a very strong correlation, so we'll see.
Definitely. You always wonder what will be in the healthy condition. But
right.
It's very hard, but...
Yeah, definitely. I think the animal work is an exciting avenue for that, obviously, because we can't implant healthy people, but... and I guess also the longitudinal aspect, that's one question I had too about the non-human primate work, is we can't really record these signals beyond the operating room or in the externalized setting. So if we could have some more longitudinal data to see how it changes over time and its stability, I guess, with DBS, I think that'll be an interesting future direction.
Thank you. So Enrico, you are approaching something that I think is very interesting, which is the asleep response of DLEPs or ERNA. In my opinion, ERNA is a subset of DLEPs, but we can discuss that outside. In any case, my question is, DBS surgery in asleep is something that has always been puzzling many. There are some neurosurgeons that do it, but there are some neurosurgeons that don't. Could DLEP be the biomarker to guide asleep surgery or support that? What are your thoughts based on all that you are doing and your findings?
Yeah, so it's a really good question. So we are also exploring the fact of, so the data actually seen here actually is from DBS leads, so it's not actually using, for example, the anemia probe used during the sleep case itself. But the idea here is that, and that's why I was actually showing data across multiple locations, is that we have specific information, we can actually try to pinpoint data. The problem is that right now we are focusing, because of the sampling, on the to be within the SDN or in the environment area. So you still need to have a little bit of imaging pre-planning to see your initial trajectory. But the helpful part here is that during, for example, MRI cases, typically you cannot validate especially if the location indeed is the correct one, even if it actually has good performances. But these actually instead will allow you to basically to have information basically for closing if it indeed actually has a good correlation afterwards, post-operatively or not. And because again, when we do imaging based, we still assume partially homogeneity across patients for what we target. So in these ways that we can actually get information. So more to your question is, I do think that these can be actually used just straight up for guiding essentially where to leave the lead itself, not just programming actually afterwards. And this is the reason why. At the same time, actually, the helpful part is that interestingly, they're still elicible actually, even under different anesthesia. We actually tried with sevoflurane, propofol, a mixture of the two, a couple other ones. But so the point is that we don't need to use, for example, use ketamine as it's used for recovering beta or actually all the biomarkers. So I think it's just interesting for that aspect.
Yeah, I didn't notice your signals were about half the amplitude when they were in awake, so that's a challenge, but thank you.
All right, so while we're getting the next person on the microphone to ask another question, a follow-up that I will have quickly would be, is there any work on trying to estimate ERNA to, or DLAP, I guess, so how close is too close to the sweet spot? And is there any relationship between the distance to borders, right? Because it's pointing towards, well, this is the contact that we need to use, and that's, no pun intended, no-brainer, in those situations. But, again, this doesn't help. I mean, it helps us with a starting point, but sometimes, the place where you see a lot of physiology coming from may not be the ideal place to stimulate because it might be too close, let's say, to internal capsule or to something along those lines, right? So I don't know if there is if there are any trends, towards helping us identify not only the best depth in terms of the electrode, but also on the medial to lateral aspect of the target.
Yeah, definitely. So I mean, part of the work if you want to show as well for directional, but at the same time, the 3D reconstruction as well, you were showing here afterwards. The idea is to actually basically get not only that information, but actually indeed like, in 3D coordinates, where are you, basically. So that's one of the questions. So we think there is actually spatial information we can actually record from that. Aside from like just having factors of volume conduction, like from the, what's supposed to be the hotspot, the generative D-lab itself, there's the question of like how really well contained actually that section that really generates this activity or not actually starts working with. So there are multiple aspects basically that I think that are encoded there. But yeah.
Yeah, I think there's additional analyses that we can do as well of correlations between ERNA and side effect thresholds that haven't been done yet. And so if we could use ERNA to predict what those side effects may be, then that may give you some information about medial lateral and relative to the internal capsule, but to be determined.
Awesome. Is Dave asking a question, or is that our time to leave?
Yeah,
okay.
time to leave.
Okay. All right, thanks, guys. It was great.